I'm Levine from Rice and the Center for Theoretical Biological Physics, and I'm going to tell you about some work on directed cell motility, where cells use chemical information to decide which way to go. You heard a little bit about this in some of the student talks on Friday from both Rice and actually from Maryland, and I will talk about some already published older work on single cells, and then some newer work on single cells, and then on some very new work on asking about, can clusters of cells, can cells have collective guidance capabilities that go beyond what they can do as individuals? Okay, so let's get started. The basic phenomena of chemically guided motion of cells is something which occurs in a wide variety of biological cells and biological systems. Here is an example of neutrophils, white blood cells that are in the blood system, and then you create a wound, and those cells are able to sense chemicals that are being emitted by the bacteria in the wound, and they then bias their motion. They leave the blood system, I guess I can use this, they leave the blood system, going here, they head downwards and make a beat, make tracks directly for the wound, where of course they're needed. So the idea of chemically guiding signals for biasing the motility machinery is well known. It applies to the immune response, it applies to wound healing, it applies to cancer metastasis, and many other conditions. Now we're going to study this in a simple organism called dictyostelium. You heard a little bit about this on Friday. It's an amoeba, which is a eukaryote about 10 micron size. Here is a dictyostelium cell moving in a microfluidics chamber in response to a chemical gradient. The gradient is obviously going from left to right. And you see directly this bias of the motility machinery. The red bursts are where actin is actively polymerizing, and this is known to push the front of the cell forward. And the green here is myosin, which is where you have contractile bundles form, and that sort of lifts the cell off the substrate and carries it forward. So the idea of directed machinery is that the chemical signal that comes into the cell, that's detected by various sensors, we'll talk about that in a moment, is used to decide that this end is going to be the front, and this is where actin is going to polymerize, and this end is going to be the back, and that's where myosin is going to bundle the actin and create contractile units. So when you look at that in more detail, you can begin to see the process whereby the external chemical information is systematically transduced into signals that go and control this cytoskeletal machinery. So for example, this is a work of Paran and Gabriolis, who in some sense started this field of thinking about how this might work. If you look at where the receptors are, cells, dictyostelium cells detect the chemical, in this case it's cyclic AMP, via G protein coupled receptors, and those receptors are uniformly distributed on the surface. So here's a cartoon version, and here's an actual fluorescence picture of GFP fused to the cyclic AMP receptor, showing that the cell has uniform detection capability. Of course, when you put the cell in a gradient, then you get slight differences in occupancy, ligand binding of those receptors on opposite sides of the cell, and that's how the cell biases its motion, and the cell translates small differences in occupancy to large differences in its biochemical state at the front and in the back. So for example, here is some particular kinase, the PI3 kinase, and the PI3 kinase becomes active only at the front of the cell. A phosphatase, which actually opposes the action of PI3 kinase, P10 phosphatase, becomes active at the back of the cell. RAC, which is something which eventually causes the polymerization of actin, it's sort of a molecule sort of sitting in between the receptor pathway, eventually going towards actin dynamics, becomes active at the front of the cell, and myosin, which you actually already saw in the previous movie, becomes localized and active only on the back of the cell. So the secret of how the cell takes the external information and creates its guidance is that it creates a different biochemical state, a state of frontness on one side of the cell, where a whole bunch of reactions are in some particular biochemical state, and a different biochemical state at the back, a backness state, and the frontness state will eventually drive the actin polymerization, and the back state will eventually drive the contractile. Now, we're going to study this using both experimental Linux-based experiments and also sort of theoretical models. And the one thing that we wanted to, before talking about, is I'm going to show some experiments. And when you have experiments, you have to pick some member of this to be your molecule that you're going to track and try to understand. Sort of what data are you going to have available to test and predict in your model. So we focused on one particular molecule that's part of this pathway. So here is the pathway, at least in some reduced version. Here is the receptor, CAR1, cyclic AMP receptor number 1. This eventually goes through a molecule called RAS. RAS is a very famous molecule, not because of its role in mycelium, but because of its role in cancer. So we're not going to talk about that here. But in mycelium, RAS is used as a sort of switch point that eventually takes the signal from cyclic AMP and eventually, through some bunch of intermediates, drives the active polymerization and also drives the signaling, which I'm not going to talk about. So here's a picture of RAS, and one of the reasons, and there's sort of two reasons why we're focused on RAS. One is it occupies a sort of very central point in this pathway, very early up, so it's a good indicator of what's going on. And the second reason is that it turns out you can attach a fluorescent marker to a RAS binding domain such that RAS will light up when it's been activated. So you can track in real time this signal that's going to be used to go from the chemical external signal to the internal motor driver. So here's a picture of the same cell, same setup, but now we're seeing RAS activation instead of seeing actin. And what you see here, notice is there are bright patches, again on the front of the cell. Again, the gradient is from left to right. This is a picture of the device. And again, this is just an indication which you can quantify that the RAS brightness, the activity of RAS, is a good indicator as to how the cell is going to move and where actin is going to polymerize. So I said you can do that quantitatively. So this is going to be the type of data that we have available, cell motion, plus information about the internal signaling monitored through the fluorescence of this RAS molecule when it becomes active. Okay, so let's talk. So I talked a little bit about the experiment. Let me talk a little bit about the theory. And the question for the theory is, why is the theory interesting, or why is the problem hard, or at least harder than many other problems that one has faced in the signal systems biology realm? And the answer is because the cell is faced with a challenge which is a little bit more difficult than the normal challenge that it has to face. Normally, when you talk about signaling, when you write down all these systems biology models, you're talking about a cell responding to an absolute change in concentration. And absolute changes in concentration directly correlate, if the cell has the right receptors, to absolute changes in the number of bound molecules on its surface. And you can easily imagine how that gets fed into the interior and causes various downstream biochemical things to happen. When you're sensing a gradient, though, you have a challenge. The challenge is easily thought of sort of in a hypothetical experiment of here is one side of the cell, and here and that side of the cell has a bunch of occupied and unoccupied receptors. And now imagine that the cell has learned that that side of the cell has been subjected to 100 nanomolar concentration of cyclic AMP. And now you say, should I become the front? Should I turn on all those reactions that make me a front? Or should I turn on all those opposite reactions that make me a back? And if you think about it for a moment, you realize you actually can't decide. Because it isn't enough to know what your local concentration is, which is 100 nanomolar. You also need to know what's going on the other side of the cell. Is the other side of the cell at 90 nanomolar, in which case you should be the front? Or is the other side of the cell at 110 nanomolar, which means you should be the back? So you need integrated information from around the cell to sense the gradient. You need some spatially extended way of processing what's going on. You can't just do it by making a local point measurement. You can't just do it in language of physics. You can't do it by basically having an ODE type model. You need to have some type of spatial communication to give you this comparison between things that are happening on different sides of the cell. So this was realized very early on and led to a general concept of how you would build models to accomplish this. The general concept is called LEGI. LEGI stands for Local Excitation Global Inhibition. It was actually proposed first by the biology experimentalists and then was quantified in some sense by Iglesias and Levchenko from Johns Hopkins, and has been developed in a number of different directions since then. But the basic idea of the Levine model is that you have two processes appearing in parallel. You have an activation process, and here I've sort of labeled my cell, this is a schematic of the Levine model. You have an activation process. So imagine the cell is in a chemical gradient. The external chemical is high here and is low here. So these green molecules represent the activation, the excitation part. So, you have 4 levels, units of 4 excitation here, and units of sort of 1 or 2 excitation in the back. And then you have, at the same time, going on in parallel, an inhibition process. And the inhibition process, again, creates inhibitors, create more on this side than on that side, but the secret of the model is that the inhibitors don't stay at the membrane. They rapidly diffuse throughout the cell. They diffuse sufficiently rapidly that they, in some sense, average out to a uniform concentration of inhibitors. So here, the inhibitors, although they were made preferentially on one side, in fact, very quickly adopt a uniform concentration throughout the cell. And now, if you just subtract, if you look on this side, you have 4 activators, 2 inhibitors. That gives you a signal of +2. On this side, you have 3 inhibitors and 2 activators. That gives you a signal of -1. So it takes the gradient and it converts it. This model takes the gradient and basically uses this inhibitor field to zero out the average value, and then the residual signal in the Levine model is above baseline on the front end and below baseline on the back end. Now, the simplest version of the Levine model is really very simple. There's, I said, all sorts of various bells and whistles you can add to make it more biologically realistic and deal with, and I'll talk about one in a couple of minutes. But the basic strategy of the model looks very simple. You have this activating field, which, of course, decays but is driven directly by the external signal. So S is the external signal in terms of bound receptors. You have an inhibitor field, which lives everywhere in the cell and diffuses. It's created on the surface by the bound receptors, but it doesn't just live on the surface, it diffuses everywhere. And then you have this downstream effector, which reacts in a positive direction to the activator, in a negative direction to the inhibitor, and this is the final signal that goes, and acts downstream that drives all the rest of the circuit. In terms of our experimental setup, we identify what's going on. The S is, of course, the receptor. We know what that is biochemically. We have the effector is the activation state of RAS. That's what we're actually going to measure as our effector in our experimental system. And the pathways of activation versus deactivation of RAS. RAS is a small GTPase molecule who gets activated by molecules called GEFs and gets deactivated by molecules called GAPs. And you don't need to know anything about that, but you can identify specific molecules that will both activate and deactivate this RAS molecule as part of our simple network. So the first thing we wanted to do, so this model's been around for a long time, but it never really been, in our opinion, tested in a sufficiently quantitative manner. So we wanted to test this. It turns out there's a very simple prediction you can make about what this circuit will do, which is that this circuit actually perfectly adapts if you don't give it a gradient, if you give it a uniform signal. Why is that? Well, I argued that the role of the inhibitor is to cancel out the mean value of the signal. Well, if you give it a global signal, no spatial gradient, the mean value is the local value. So the inhibitor cancels out the signal and the system doesn't do anything. Now, it doesn't do anything in steady state, but the activator is faster than the inhibitor because the inhibitor needs to spread out, it needs to, it has slower dynamics in general. And so what you get is you get a transient response, which then returns back to the original baseline if you give the system a uniform stimulus. So we design, we in this case means really Alex Royceman at UCSD, design a microfluidics device to very carefully give the cell very carefully controlled pulses of cyclic AMP, and you can measure things very carefully. Here is some typical data from this. The down is the signal just the way it's defined, and you get this transient response, and then everything returns to baseline. Here is the final steady state value. This one is probably within the error bars, also returning to baseline, which you might worry about it, but up to all the others, clearly over 4 orders of magnitude of size of the signal is perfectly adapting to the cyclic AMP concentration. So it turns out that in a very, very nice paper by Chao Tang and Lim, they pointed out that if you know that your system is perfectly adapting, and you know that there's some core circuit that's trying to accomplish that, then in fact you can write down only a couple of simple models that actually succeed in doing that. Here's sort of basically 2 up to small changes. There's the one I pointed out here, where you have this competing pathways, and the one here where you have feedback, an integral feedback. This type of circuit is well known because this actually is responsible for E. coli chemotaxis and the way that system adapts. But here we propose this. It turns out that if you look carefully at the kinetics of the system, you notice here in the graph that the different strengths all adapt, but the timescales of adaptation are different. And it turns out that these models behave differently in terms of how the timescale of adaptation depends on the strength of the stimulus. And the result here, which shows that the higher stimulus returns more quickly to the baseline, is only consistent with this class of models and not with that class of models. So essentially what you can do is you can fit all your data, but even without fitting the data, you can make a decision about which class of models is relevant just by understanding the basic structural predictions of each of those models. And then once you have something which agrees in broad structural terms of all the trends that you see, then you go through the exercise of fitting all the parameters, fitting all the data. We did all that, and this was published in 2012. Now, a couple of bells and whistles. One important bell, or whistle, depending, is the fact that the Levine model in its bare bones version zeroes out the mean, gives you a plus and a negative, but it doesn't amplify the external signal. I already told you that one of the secrets of gradient detection is you go from a small difference. The typical gradient in those experiments across the cell is maybe 5 to 10%, and yet you're making a 100% change when you decide this is the front and this is the back. So obviously part of that has to be to amplify the external signal, and you can do that in the Levine model in a number of ways. One way which we proposed was to make the effector equation ultra-sensitive rather than having it be just linear kinetics, and then with the right balance condition, you can actually get a very large amplification of the basic Levine mechanism. So we think that this is actually the simplest way to get the type of experimental data that I showed before. Now, let me talk about so this is sort of work that has been ongoing. Our last work on this was published in 2012. I want to talk about something which is new and then something which is very new related to what's going on here. So let's start with the thing which is new. So by new means that we actually published this last fall, 2014, which is we asked the question now experimentally and also theoretically, does the cell have any memory of what's going on? So previously, all our data was the cell is sitting there, and you give it a stimulus. You give it a constant stimulus, or you give it gradient, and you show you that data. You can fit all of it using the model I described. But you can imagine, let's say the cell is sitting in gradient for a few minutes, 10 minutes, and now you switch the gradient. Does the cell have any hysteresis? Does it remember that it was sitting in a gradient for 10 minutes, or is the cell, every few seconds just making a de novo decision? So the way you can do that experimentally is by, again, designing a different microfluidics device that takes a gradient that looks like this and very quickly makes it into a flat signal. But of course, when you do this, you have a choice. You can make the signal flat at 0, you can make the signal flat at the top of the gradient, or you can make, as in this picture here, the signal flat at the midpoint of the gradient. So you have a continuous variable of what mean do you set the mean to after you rapidly switch away the gradient. And it turns out, so here's now a picture, this is what happens if you switch it off completely. You start from the gradient, and you completely get rid of the cyclic AMP concentration. So here's a pictorial representation of that bright RAS spot. What you see here is this red is the signal, the RAS signal, and the cell membrane is now been wrapped to this direction over here, and this bright patch in the middle shows that the cell is putting RAS in the front of the cell. And so at time 0, you switch off the gradient, and the RAS signal goes away. However, if you change this, make it constant to, let's say, hundreds, and keep this at the top, then the RAS signal doesn't quite go away. It weakens a bit, but you still but less strongly. So the cell has some type of sort of strange memory depending on exactly how you switch the gradient. And this can be done more extensively in detail, and this was described in this paper. And that, of course, requires an extension of this Levine model to try to explain this data, because the Levine model on its own cannot explain memory that lasts for minutes. The Levine model's dynamics is over within 10, 20 seconds, so we can't do it. So what we did is we took the basic Levine model, we coupled it to a bistable switch. Here, this is a memory module, and the details are not very important, but now when the cell is in a gradient, if the gradient is strong enough and lasts for a long enough time, it flips something which is a switch. That switch stays flipped even when the gradient goes away for at least some amount of time, and now you can explain the data. This is typically what the data looks like. And so when you go to 0, the signal goes away completely. When you stay at 50 or 100, the signal stays. And now this is a model calculation. When you go down to 0, the inhibitor is enough to immediately flip you out of that state. But when you keep the concentration high, at 100, which actually now is a higher mean cycle AMP concentration than you used to have before, that actually keeps the cell with this memory module active, and you actually get a continued signal. Now, why is this important? Well, cells, and we heard about this a little bit on Friday, cells have a challenge of using this system to actually detect waves. If you detect a wave, it means that you have to distinguish between the rising part of the wave and the back part of the wave. And so if you don't remember anything, if your response is immediate, then, okay, you have a wave come at you, you see the concentration is rising, so you walk this way, and then the back of the wave hits you, and you see the concentration is falling, and you walk the other way. And on average, you don't get anywhere. So this memory of the system is connected to the cell's ability to distinguish between fronts and backs of waves spread apart by some amount of time. So we actually did that experimentally by creating a wave generator in the microfluidics device. Now we send waves to the cell, and this is what the data looks like. So let's start at the 20 here. If you look at 20 minutes duration, so you give the cell a front of the wave, and then 20 minutes later you give the back of the wave, then the cell moves forward here and moves backward here. So this is a measure of the cell motion now. The cell up here means it's moving in one direction, down here is moving in another direction. By the time you get to a 6-minute wave or an 8-minute wave, what you see here is the cell is moving forward in the front of the wave and continues to move forward even in the back of the wave. So the cell is remembering and essentially ignoring the back of the wave if the back of the wave comes soon enough compared to the time scale of this memory module of the system. And again, we can directly model that behavior in our model. And we think that during early aggregation, when the cell is first deciding which way to go and is being hit by waves in a variety of directions, this may be a relevant mechanism to help the cell sort of make a choice. I'm going to go towards that center and maybe not to some competing center, and this method could be very useful for us sort of doing that. We're trying to test that now directly with both experiments and theory. Now, in the last 3 minutes or 4 minutes, what I have to say, chemical guidance is not directly being sensed by receptors at the individual cell level. It is not the only way individual cells can bias their motion. This is a cartoon from a recent paper by Levchenko's group showing that cells can bias their motion by other methods. In particular, it's funny because you see this fish, and you have to keep remembering that cell, that Dickey, when people use this as representation from Dickey, this is the front end. So it's sort of funny. But nonetheless, what we see here is we see a competition in this paper between individual cell chemical guidance and the fact that if there's another cell in your way, there's a mechanism called contact inhibition of locomotion, called CIL, which says that you basically are repelled by another cell. You don't want to move into another cell, so you're repelled. So if you have two cells like this in a chemical gradient, the chemotaxis can push you this way, but the chemical, you might want to go the other way because of other cells. So this is a well-known phenomenon, and actually we propose that this is responsible for some recent experimental data, which is quite remarkable. So here's a movie. This is now not epithelial cells, it's actually lymphocytes. But what you see in this movie is a gradient from top to bottom. And if you watch carefully, you'll notice that single cells don't know where to go, but large clusters are all moving downstream. So here is the system where clusters of cells, and the clusters are not huge, 20 cells, 10 cells, whatever. Clusters of cells seem to be able to use the chemical information to guide their motion, but individual cells are not able to do that. And curiously, what they showed in this paper is under some conditions, it's not only that you can actually find conditions where single cells will go in the opposite direction of the clusters. So the simplest interpretation of this, we might have thought, was that individual cells, are very lousy, sort of weakly biased by their motion, sort of crappy detectors of directions. But if you add 20 signals together, you just get an averaging out, and so you get some net signal. But this part of the experiment shows directly that it's not that case. The individual cells left on their own are not doing the same thing as what the cluster is doing. So we proposed a simple model, and this is where I'm going to end, for how this works, essentially based on this contact inhibition of locomotion. We have cells together in a cluster, cells move according to some simple law, and the only input to this model is that the cells repel each other when they're next to each other. And now, if the coefficient of the repulsion term is sensed by the cells, and the cell biases how much contact inhibition of locomotion there is based on the chemical gradient. So this is each individual cell is measuring the concentration, not measuring gradient, but it's using that concentration to decide how sensitive it is to repulsion. Then you can prove that this equation will give rise to gradient directed motion of the clusters with some simple gradient law, and you can work this out analytically. We did that in detail, and I'll just show you one simple simulation of that and quit. So here is just, not going to be serious, but here is sort of typically what you see in that, so similar to that, whereas if you look at this, these individual cells are going nowhere, but once you form these small clusters, those small clusters can systematically move downhill because of the way their repulsion from each other is biased by the chemical gradient in the system. So we propose that this is a new collective sensing strategy that goes beyond what individual cells can do, and we'll be predicting and we'll see how well this agrees. So I'm done. 1 minute more than I hope. Chemotaxis is a fascinating example of cells acting upon information extracted from their environment. So it sort of falls in the general area of understanding how cells are able to do functional behavior by measuring what they need to do. We've worked a lot on extensions of this work to the actual motor parts of the protein. I haven't talked about that at all. And the new part of this, which is under submission somewhere or other, is that multicellular systems may have their own form of chemotaxis that are truly collective, that do not depend on the ability of single cells to detect gradients. They depend on single cells detecting concentrations, and then the whole thing together is able to create collective guidance. So with that, thank you very much. Happy to take questions.
In the CIL-based collective motion dynamics, is that dependent on the overall density of cells?
Well, in principle, yes. So in other words, the way we've done it is we, in a weak gradient limit, what happens is the cell forms a cluster, and there's some equilibrium in some sense between the clusters and the gas and particles around them. But the cell forms a cluster. That cluster is more or less constant density in our model, because the distance the cells stay apart from each other is determined by the strength of that repulsive term. So we don't have, so in our model at least, and in the experiments, as far as I can tell, the clusters typically are variable in size but have a relatively fixed density. So we don't have an answer to that question because our model always gives a relatively fixed density for the clusters.
Is there any minimal overall densities to form clusters in the first place?
Yeah. So you mean so I didn't talk about forming clusters. Yeah. So in other words, there's some type of equilibrium in a sense between the forming clusters and the individual cells, and putting more cells in the system will create bigger clusters. So yeah, but at the level of, so maybe I misinterpreted your question, but at the level of each individual cluster, that has roughly a fixed density in the system.
Hi, I have a question about the beginning of the talk. So at the beginning of the talk, you had, for many components, like you had a physical entity that was the component, and then you introduced the hypothetical memory module, and I wondered what your thoughts are on what that actually might be.
Yeah, so we don't know. I mean, so we work, I mean, the only clues we have a few clues as to where to look, but I don't have an answer to your question because I don't know. If you knew, I would have told you. The clues we have is that in terms, so one thing dictyostelium does, which is sort of smart, is that it doesn't turn on the chemotaxis system until some amount of time. In other words, it doesn't have this system when it doesn't need it. Dictyostelium, when the food is plentiful, chemotax doesn't send out waves, and therefore doesn't need these proteins, so the gene expression of the components of the system are time-dependent, they're induced by the chemical signals. So that's a separate story, but it turns out that this memory module seems to be induced later. So we can narrow down what genes it is by looking at what the time course of induction of various pieces are. So when we do that, we came across a variety of genes that look like they're sort of cyclic GMP-dependent phosphodiesterases and things like that, which may be candidates for this. And in the lab, they're busily trying to knock out those particular things. And the home run would be creating a mutant that does chemotaxis to static gradients, but cannot do the memory module. And we have a few leads of that, but nothing that we really think is really it. So the short answer is no, but we're working on that. The people in the lab at San Diego are actually working on that.
Thanks a lot for being here. Go on.
Okay.